So hello students, and welcome to week 7 of the next generation sequencing course. We talked in the last lecture in quite some detail about the sources of error in SNP calling and genotyping in next generation sequence analysis, and also talked about the need to filter variants in order to develop the highest quality call sets. Today we're going to shift away from a little bit of the sort of more theoretical side of why we have errors and the need for filtering, and into sort of the more practical side, that is the bioinformatician. How do we go about handling and processing these large SNP data sets? And so that's really the subject of the homework as well. We'll be having you work with VCF files, working with both tools in the Genome Analysis Toolkit, or GATK, as well as VCF tools. And then I'll also introduce you to what I think for many of you will be a welcome sort of opportunity to work with sort of a graphical interface to visualizing data and data display. And we'll be using what's called the Integrated Genome Viewer for that, and I'll introduce you to that at the end of this lecture. So we talked about in the previous week 6 lecture series the need for filtering, and I introduced you to the idea that when we're working with non-human data anyway, we have sort of a large range of potential filters that we can apply to data. Some of them are arbitrary, as best as I can tell, and that we have some ability to detect or use filters that might be improving our call set, and we can use things like the transition transversion ratio to tip us off as to which sets of filters may be actually removing false positives, as to in contrast to just basically making our data set smaller. So this was a study published, I believe, in Nature on Plasmodium. This is a set of variant filters that they applied, including basically that they just decided that they were not confident at all in calling SNPs outside of coding regions. They used biallelic SNPs only. Those are SNPs that where there are only 2 alleles segregating in the population. They used missingness and as a criterion, that is, if a certain number of samples had more than had a certain certain number of samples had missing data, then those sites were considered to be enriched for false positives, so they excluded those sites, et cetera, et cetera. But I also wanted to understand that even though in human data where we have true positive sets, such as the HapMap project, so this is array-based genotyping in some cases, or other genotyping strategies where we have much higher confidence in the genotypes themselves. These are what we call our gold standard, or DB-SNP, or SNP databases that we generally consider to have very low error rates. And when we have that type of resource, we can use that to infer the aspects of our unknown call sets, such as the false positive rate, which can lead us to identify certain filters that might help in our SNP calling. So we had these, I introduced to you the base quality score recalibration and the variant quality score recalibration, which are sophisticated methods that are aimed at reducing false positives, but I also didn't want you to leave feeling like, if you're working with human data, that you don't have to apply filters. So here what I did is I just went to the 1,000 Genomes project, into the supplementary material, which, by the way, I'm going to post on the course site, simply for you to kind of glance over to sort of get a sense of the thoroughness that they're applying in the human data sets. This standard is very high in human data, and I want you to at least come away from this part of the class appreciating that. And so here were some of the different filters that they applied in the 1,000 Genomes Project paper. Again, things like depth of coverage were important. So in cases where depth was too low, they would remove a site, or in cases where depth was too high, they would filter a site. So recall that too high depth can reflect structural variation, where you're getting paralogous reads mapping to a single region in the reference genome. So that is, duplicated regions in a sample can map back to the reference genome to a single unduplicated region and have, in principle, twice the coverage depth, simply because of this duplication in the sample. So in any event, these were some of the filters that they applied in the 1,000 Genomes Project, and in many of the cases, in all of these cases, these are not arbitrary sort of thresholds. They were able to take the HabMap data and really identify the filters and filter thresholds that are, reducing their false positive rate by a certain known amount. Okay, so how do you filter data in the VCF sort of format? So essentially there's this column we haven't talked too much about here, this filter column, okay, and you can see in this particular VCF all of these four records, or these four variants, are passing filter. You'll also note that by default in your call sets that you generated in week 7 that there is a dot or a period in each of these positions here in the filter column. That is because no filters have been attempted to be applied yet. So dot just means missing data. So the way essentially filtering works is that we apply a filter that is some threshold where each read is each record or each variant is evaluated relative to that condition or threshold, and then the pass, if a read did not meet this particular condition, excuse me, I keep saying read, but I mean record. If the record did not pass that particular condition, then the name of the filter that you decide to apply would appear here in the filter column. So these filter tags, as I'll call them, are only applied then to the records that do not pass that filter. And as you'll see in your homework, that certain if a record does not pass multiple filters, then a semicolon separated list of filters will appear here. So the names of the filters will appear here, listing all of the filters that particular record failed to pass. So typically we filter on the quality column, and we can filter on any tag in the info column, and we can either do conditions such as the record must be above a certain quality, or you'll see here various things such as depth, which unfortunately this example VCF does not have, but the DP tag is a common one and is present in your SNP call sets. And that DP tag records the total depth across all samples. Remember, most VCFs are multi-sample. Yours is just one to keep it sort of simple and small. But we record multiple sample genomes in a single VCF. I'll come back to that later. But in that case, the depth tag would include the sum of the total number of reads that map to this position across all genomes. And if we apply a filter that says that the depth must be, say, greater than 15 across all of our genomes, then if it does not pass filter, then a tag would get inserted here for the filter to indicate that record did not pass filter. So it's fairly standard to actually leave these filtered SNPs in the VCF. However, sometimes for convenience, you physically want to remove the records that did not pass your set of filters. And for that, we have a set of downstream tools in which we can remove those those reads that don't pass filter. And we also might rely on third-party software to provide us with the option to exclude records that did not pass filter. But generally speaking, sort of the best practice is to leave those filters in place. You don't actually reduce the size of the VCF. We leave them in there, and then cognizant of that fact that the filtered records are still there, we go and proceed with analysis and make sure that filtered reads are excluded in certain analyses that we want to exclude them. Okay, so how do we go about applying filters to VCF? So this is a very applied part of the lecture series here, where I'm giving you some tips to hopefully save you some time as you're doing the homework and in preparing for generating high quality call sets. Essentially, variant filtration, this has a white space here. It's actually all one word. That is the name of the program that GATK uses to apply filters. And so what we do is in the variant filtration program, you can look up if you just Google variant filtration and GATK, your first hit will probably be the documentation for this. I point this out in the homework where I give you the link there. But the two arguments that always come in pairs, okay? They can be specified multiple times. That is, a pair of these arguments can be specified as many times as you want in a single command line to indicate multiple filters that you want to apply to your VCF. We have the filter name. That will appear as a tag in the filter column for any record that does not pass the filter. Then we have what's called the filter expression, and that's what I sort of outlined below here, is how to define a filter expression. So this gets into a little bit of things that I certainly had hoped to avoid when I became interested in bioinformatics, but something obscure like the Jexel Java expression language is the language that's used to evaluate certain conditions at the command line. And so here we have an example. Let's say that we want to apply a filter to variance with an MQ greater than 10. So MQ is I won't go into exactly what that is. It's the RMS mapping quality. So this is from the info header line in your VCFs that you generated from your call set from week 6. You'll see that the info, so the info column has MQ tags in it with values, okay? And these values, the thing I wanted to point out is that how you define the expression, okay, is actually critical as to whether or not the expression will be, or the condition will be applied to your filter. So here I want you to notice in the header line where we have a description of each tag in the info column, okay, you'll see that you have type equals float in this particular example, whereas in other examples you'll see type equals integer, or others might be something else, but these are two examples. And if type equals float, what that means, this is a decimal floating point number, and what this means is that there's a decimal here, okay, and that you have that the values, okay, you'll notice in the info tags will have decimal floating points, and if they do, then you must specify your criterion with a decimal floating point. Okay, so here that just means if we want to apply this criterion MQ greater than 10, but you must which means that we are going to filter records that have an MQ greater than 10. So we'll apply a filter if this condition is met. Okay, that's critical to understand, that we're applying the filter to records in which MQ is greater than 10. But notice here, we're not just going to specify MQ greater than 10, where 10 is an integer. We have to specify a float, okay, because that is the type of number that the MQ tag has associated with it. So applying a 10 here without the floating point number would lead to a problem. Okay, so you have to check in the info column when you're defining your filter expressions, you have to check whether or not the tag in the info column is either float or integer. Okay, so then we can apply whatever filter name we want to associate with that expression. Okay, so here we're calling it MQ10. So any record that had a MQ value greater than 10 would then have the MQ10 filter applied to it. Okay, so here I'm just summarizing. Specify condition with a non-integer because type equals float. Okay, so let's look at another example. Here's one where the type is integer in the header, and here we have MQ greater than 4. Okay, so this is what notice the matching between the integer here, and now we're expressing our conditional here, MQ greater than 4 as an integer as well. Okay, and just notice that these are in quotes, that's double quotes, that's important. And here, oops, I think I should have renamed this. This is a typo. I just copied this over. This should be MQ0, GT4 or something, whatever we want to name it. Okay, so you can name this filter name whatever you want. So let's look at an example where type equals string. So here in the info column, here's this. This is a tag called snipf effect, and I introduced to you in the last lecture the idea that you can annotate snips with their effect. So for intergenic snips, okay, what we might want to do is say, let's say we want to identify, only snips that are in intergenic regions, because while we're not filtering it in the strict sense, well, remember this one study, the falciparum study, they actually excluded all intergenic snips as well as intronic snips. And so in this case, a simple way to sort of, execute that on a large data set of, say, 10 million snips, you can simply, apply a filter to indicate that you want to exclude or filter any, snips that are in intergenic regions. So notice here that this is string equality, which is actually in the Jexel expression language, is actually the same as numeric equality. Okay, I think this is pretty clear, so let's move on. So I think I mentioned already that variant filtration, that's the program in GATK that applies these filters. You can apply these pairs of arguments as many times as you want in a single command line. I also wanted to mention that there's other cool sort of like functionalities here where you can input sort of a set of intervals. We haven't talked about bed format yet. We should be doing that probably next week, but this is just a set of intervals, say in the genome, that you want to mask or exclude or filter any SNPs in the interval specified in this repeats bed. So this is something that I apply in some of my own data where we have, we expect the false positive rate to be higher in regions that are repetitive simply because the mapping of the reads are more likely to, be errant. Okay, so mismapping of reads in repeat regions. So what I do is I apply sort of a global mask where I remove variants or filter variants that are in a set of intervals that correspond to known repeats. And for shorthand, my filter here, so mask name is, this is simply taking a single file and applying for all intervals. It applies all SNPs in those intervals. It gives them the reps filter, again, in the filter column. Okay, so here's a case where, from some of my own data, where I've just taken a section out of a VCF file here, and here's a region of the genome that has actually a lot of records that are filtered. And you can see here that there's a list here of multiple filters, indicating low quality, that the quality is less than 30, that a genotype was heterozygous. This was in a haploid call set, so I expected no heterozygous, so I actually filtered out those SNPs that were heterozygous. And then, of course, you can see those SNPs or variants that pass filter. After you've applied variant filtration, it will, by default, apply pass to this column for records that passed all your filter conditions. So sometimes, though, you do want to actually physically remove the filtered variants from your data set. I do this sort of at the final stages of analysis where I only want to be working with my SNPs that pass all filters. And in that case, I do go ahead and remove the SNPs that did not pass filter using this option here in the exclude filtered in the select variants program. So here again, remember, just a reminder, when you guys are executing GATK, we always specify the GATK path to the GATK jar file. We have to specify the reference always, and we always have to specify the program, this case select variants with a T option. Frequently are specifying the variant, that is the VCF, the output VCF in this case, and here I'm applying a couple of just arguments that sort of lead to different output files. Here, this excludes any variant that was filtered here or did not pass filter. Here I'm restricting the type of variant to SNPs only. Remember, in week 6 you also called indels in a separate sort of VCF. You had indels, but you can also combine VCF so that you have SNPs and insertion deletions in the same VCF. So here you can exclude any insertion deletions or other types of variants that might be present using an option like this, and then we can restrict alleles to biallelic using this syntax here. So another thing that is very useful, here again, sorry class, I've made a typo. I meant this is actually introducing a tool called the variants to table tool. So this should say variants to table here. So how do you create a table from a VCF? Because why do we need to do that, first of all? Well, the reason is because VCF is a very rich format with a lot of information, and it is difficult to parse the VCF data. That is, to extract data for individual tags. So remember, for example, remember I mentioned the information tag called DP, which is the depth. So here you have a string of information in the info column, and you want to extract only information from depth. So how do you go about doing that? Well, it's actually quite difficult to write specific code that will take care of all of the sort of the vagaries of VCF format, so to speak. So there's different types of tags that can be added to the info column. The info column can be actually quite difficult to parse. You have to know the format very well, etc., in order to do that. So instead, what GATK developed was a simple command called variants to table, which would simply dump all of the information you want from any column into a table. And that's very convenient because now we can read that into R or whatever our favorite processing software is, and we can go ahead and treat it just as a table. So I find this to be very useful because I feel very comfortable working with tables in R and generating plots, generating summaries, doing more sophisticated analysis. So how do we do this? How do we execute variants to table? Remember, we have the -R reference path to the path to the reference genome. We have our program, which is variants to table. We have our input VCF. Now notice that we can specify the -F option multiple times. These are just indicating the columns that we want to dump, and here we're indicating the tags in the info column. So these are the info column tags. DP and MQ are two examples that maybe we want to dump. We also have here in green, I've highlighted one where we can get metadata. So variants to table will calculate certain things about your VCF that are not in the VCF originally. So it will indicate whether or not in a separate column, in this case, a column called transitions. Okay, so transitions will become a column name in your output table, and the column will contain a 0 or 1 as to whether or not that particular SNP is a transition mutation. Okay, and there's a number of these sort of metadata columns that you can have variance to table export for you. And so that you can read the variance to table documentation to get a list of those sort of metadata columns. Now, in blue here we have the genotype fields. Okay, so maybe we'll go back really quick here and just remind you that each of these samples is a different sample genome. And in the format field, we can, or the format column, we can see the subfields okay, that are in each of the sample columns. So GT colon DP means that we have the genotype followed by colon followed by the depth for that sample. Okay, so what if we want to get sample 1 genotypes and sample 2 genotypes, and we also want their depths? Okay, so how you specify this is using invariance to table. We want to get each of those as an output column. Here I've used some other subfields other than GT and depth, such as GQ or the genotype quality. But if we specify GF here, this is indicating the genotype fields, or those fields, subfields for individual sample genomes. Okay, we can extract the genotype quality, in this case the depth, and the Fred likelihoods of the genotype calls. And what variance to table will do conveniently is it will create a new column for each sample simply with .gq or .dp or .pl appended. So in this case, you would end up with a table that has sample.gq, sample1.gq would be a column, sample2.gq would be a separate column. Okay, so each column now has a unique column name so that you can extract, obviously without ambiguity, the information that you want from the table. Okay, so you can review that if you'd like. I think this is pretty clear, very useful tip, and in fact, rather, I would strongly encourage you to not ever really attempt to parse a VCF file, say using Python or Perl or R or anything else, but rather to let GATK do that parsing of the table for you and to dump that output using variance to table. So I wanted to hit on another subject here that I think is probably missed by a lot of people who, when they first start working with VCF formatted files. And that is that the VCF format has some sort of sophisticated properties here. Let's take an example to what I mean. So AC, let's look at the info column for this. This is an example. We have a multi-sample BAM with 4 samples. Okay, so here notice I've got, I'm missing the format column here. I just, removed it for simplicity here because I don't need it for this illustration. So this is a 4-sample VCF. So here's our 4 genome sequences for 3 different SNPs. Okay. The thing that I want you to note is that the AC field here, okay, counts up the allele count. So how many alleles are in, all of your samples combined, okay, and that is how many, I should say, alternate alleles. In other words, how many A's or alternate alleles are in the total sample. Notice also that this was a haploid organism I was working on here, and so we don't have the conventional diploid genotypes that you've been seeing in other examples. But so here we can count the number of non-reference alleles. So we have a 1 here and a 1 here. 1 here means non-reference or alt, and 0 here means reference. Okay, so how many are there? Well, there's 2 non-reference, so that's AC is 2. Let's look at the AC equals 4. Sure enough, we have an alternate allele here, 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 and here. Okay, so those check out. Now, this gives the allele frequency, and this gives the total allele number. Okay, that's the total number of unambiguous or non-missing data. So all of these have data present for the genotype calls. Ambiguous data you might get if, say, one of the samples didn't have any reads at that position. Then GATK's SNP caller, the unified genotyper, would not have called a SNP, and you would end up, in this case, with a dot in that position where the genotype should be indicating missing data. So AN is 4. That means that all 4 genomes had data present at these positions. Okay, so why do I go into that sort of detail here? Well, the point is that oftentimes you will want to sample only a subset of the individuals from your data set. So imagine you're working on human data, and now you want to only, extract the Han Chinese samples from, say, your 1,000 genomes project, and you want to conduct a separate analysis just on the Han Chinese samples. So what you would use is select variants, okay, to extract a certain set of samples. Okay, so here, remember we've just looked at select variants, but here I'm introducing some additional functionalities of select variants. So we can specify sample name. Okay, that's one of my samples here, CR2342. So now what we're going to do is we're going to go from a 4-sample VCF to a single sample VCF. The point is that GATK's select variants tool is a smart tool. It will adjust the information to reflect the fact that, to reflect the sample content of the new VCF. So notice here how all of the values in purple have changed to represent the fact that now only CR2342 is there. Right, so the allele count is 1 because CR2342 in these particular sites has a non-reference allele, and so the allele count is 1. I also want you to note that here, when you start removing samples, some sites will become invariant. That is, all individuals will be referenced. So they'll be identical to each other, and they will all be referenced. So notice here that when we exclude this sample here, notice that 2931 was the only one with a non-reference allele. And so this sample that I extracted here was referenced in this position. And so it's not polymorphic or variable anymore. And so if we want to keep our VCF to be strictly variance only, then you would want to include this exclude non-variance option here. And so in that case, you notice that sample 2342 has now been removed. The SNP at position 14,948, the entire SNP has been removed because sample 2342 was referenced, and therefore this site is no longer polymorphic or variable. Okay, so here we've outputted then just a 1-sample VCF by specifying the sample name. Select variants, again, allows you to specify sample name argument many times as you want to indicate the set of samples that you want to be working with. So really, the point here is that you can't just willy-nilly start to cut columns, using, say, the cut command, to, downsample the number of samples in your VCF, right? Because if we just cut out these last 3 columns, okay, and kept 2342 and all of and this information here, all of the info tags here that I've highlighted would now be wrong, okay, because they would no longer indicate the data that's present in the VCF. And that's the point I wanted to make, is that the VCF, you really want to use the tools that are available, that are smart tools, when processing and handling VCFs. Okay, so this is really your only opportunity to get that point, because I don't think we come back to this in the homework for this week or at any other time. And so I really, just the take-home message once again is to don't try to parse these tools yourself using the command line. You'll see people doing that sort of, occasionally when you look at message boards, and yet you open yourself up to some, significant risk that where you're now sort of the information in your VCF is no longer accurate. Okay, so in fact, that just did remind me that one thing about VCF tools, which I'll be introducing in a moment, is that when you export new VCFs using VCF tools does not modify these. In fact, what VCF tools solution is if you modify a VCF, it simply will not export any information tags. Or if you explicitly request it, you can export the same information from the original VCF. So this is a case where VCF tools is actually not very useful. And so I highly recommend using the GATK tools like select variants because this allows you to do the manipulations sort of in a rigorous way. Okay, so it's a little bit about best practices in handling VCFs. So I just wanted to remind you once again, I think I brought this up the next lecture about the functionality of GATK, and here all I've done is I've gone in and I just dumped sort of all of the tools that are available. Some of them, just a few of the ones, have we actually begun to work with. You'll see here in the with respect to VCF manipulation, you have variant evaluation and manipulation tools. You can see some of these might look familiar, like select variants, like variant filtration, and like variants to table. But notice that there's a lot of other additional commands now that are available. And literally, what I do is I hope you guys signed up for emails for the GATK. So what happens is they send out updates. Whenever they release a new version of GATK, you get an email. It's usually about once every 2 months, and then I usually go in and check all of the new tools that are now developed because now I can do things that might have been more difficult to do previously. I have a new tool, perhaps that can do what I want more simply. Okay, so I encourage you to kind of go to the GATK and you kind of just stare at these and these tools, because a lot of your analysis, both of VCFs and in the processing of SNPs, the production of high quality SNPs, the quality control and the validation utilities are all here. And just getting familiar with them is really half the battle, because once you kind of have a plan, you can easily find the tool that will execute it. Okay, so I did want to mention VCF tools. In fact, you'll be working with VCF tools. It is still in a massively useful toolkit, despite significant overlap in functionality with GATK. And there are some tricks that it does that are that GATK can't do. I think there's many more tricks that GATK can do that VCF tools cannot. But VCF tools does have some strengths. Its broader functionality is in merging VCFs. GATK can do that. As subsetting, we've already seen that GATK can do that. And the reason I say not recommended here is because it doesn't update the info tags, as GATK does. And so that's why I say not recommended. Applying filters, generating summaries, and this is I bold this because there's some things here that GATK can do that that VCF tools can do that GATK can't. Converting VCFs to other SNP formats, I'm not sure GATK has broadly implemented that. And you can, summarize differences, say, between two call sets, maybe generated with different SNP callers or different options. You can compare them and summarize them with certain commands in VCF tools. VCF tools also has a Perl API that this allows you to, incorporate VCF tools sort of options in, Perl code. These are all functions that are at the, you can apply at the command line, similar to the GATK tools you're now becoming familiar with. So I just wanted to remind myself here by putting this here that there's not a lot of these sort of unique summary generation tools are in the section on output statistics in the command line options part of the documentation. And so you might have a look at that, because in my view, most everything that VCF tools can do, GATK can do perhaps better. except that there's a lot of output statistics options that VCF Tools has that JTK at the moment does not. So finally today, I'd like to prepare you just for a second for your homework. I think that you'll find this to be a pleasant change in or pleasant part of the course where we talk about using and visualizing data. So in many cases, it's better to stare at data to identify patterns, particularly when you have a particular gene of interest or a particular set of SNPs of interest. Sometimes working on these pipelines, it's easy to kind of lose the forest for the trees, so to speak. So it's sort of the reverse problem where we have so much data. There's kind of this black box. We don't even look at the data, and that I think can be a problem. Okay, I think that part of good next generation sequence analysis is actually taking some time to go in and stare at individual reads, at individual SNP calls, and to identify potential sources of error and to familiarize yourself overall with the data set. Not to mention, of course, that there are also many times where your boss may say, we're interested in this particular candidate SNP, which we think might be a functional SNP in causing a particular disease. We need to go in and be absolutely certain that that genotype call is correct. So how do we do that? So I think what I'll do is just quickly sort of open IGV here for you. I've downloaded. Here you can see Java jar. This is the command you'll be using to open IGV once you get it downloaded. So you'll find information on how to do this in your homework. So we'll just execute IGV here from the command line. And here you can see here that in IGV there's a number of sort of default genomes that you can work with. So these are you want to use the same genome here as you use to align your reads, whether they be RNA-seq reads or genome sequencing, as we've begun to work with. This is the reference genome. So for our case, essentially we used human B37 version of the reference, and so this is the one you'll be using in your homework. Okay, so then here you can see the chromosomes. Here's a search window where we can search a particular gene. Here's a gene called ODF2. Now this particular genome we have associated with it, the GFF, is sort of integrated in this genome so that we have the gene models. We can zoom, we even have here the entire chromosome model. The red part here indicates the 49 kilobases that we're zoomed in on. And here we can zoom out. Okay, we can see now more gene models are appearing. Here, the exons and introns here, the exons are the blue blocks, the lines represent introns. We can zoom in and we can zoom out. Okay, so then we can move along the chromosome by clicking on the arrow. This is a small arrow here. If we click far to the left of the middle point, we make big jumps along the chromosome. If we click closer to the center, we make smaller jumps along the chromosome. Okay, so same thing for the right side here. So now let's open some data. So let's load from file. Let's see, we have here, down here is our BAM file, the one of this British individual that we've been working on. This is a low coverage sample of the alignments along chromosome 11. So here, here it's by default, for whatever reason, we were on chromosome 9. I guess that's where the gene I typed in a second ago is. So let's go to chromosome 11, and I think we were working around 65 or so thousand, 65 kb. So if we jump to that region of the genome here, and then maybe skip down a bit, you'll see reads start to appear. Here we're quite far zoomed in here. These are individual reads. I hope you can see them. These ones are light colored. But we can zoom out. Now we can see the reads in this region. Now let's jump a little bit closer here. And here you can see, the pileups of reads. So each individual one of these squares is a read. We can zoom in and you can see their orientation, left or right in their orientation. We can click on an individual read, or so here what I did is I single left clicked on the read, and I get a window that sort of pops up here that gives me all the information from the BAM file about that read. Okay, so this is the exact information that you would find in the BAM, including its cigar tag, whether it was mapped or not. This is coming from the bitwise flag, information about its mated read, so it tells us about the orientation of the mate. The read here is mapping quality 0, that's why it's white. So if we scroll down here, let's jump a ways down here. Here we also have a sort of coverage depth information. So this track here is sort of up and down, indicating coverage depth along the chromosome for this particular individual. Okay, so you're starting to get a sense of it now. So if we continue to jump along here, you'll see here's a gene model here whizzing by. Here's some different colored reads. So what do these different colors mean? Well, essentially the different colors, in this case, blue means that the paired read is very close in proximity. So the insert size is only 108, and that's significantly lower than the expected size. And so IGV goes ahead and colors that read colors that read to indicate that it's an anomalous read pair. So if we click on the right here, we can see various options we can do. We can copy the sequence of the read. We can blast the read. We can do various things we can go to the mate of the read. Let's see if that works. Oh, so here it is. The mate of the read is actually right here. And that's interesting because it said that the pair of reads were mapped 100 base pairs apart, but that could be, what that probably is it's indicating from the leftmost position of the forward read to the rightmost position of the reverse read, and that entire span is only 100 or so bases. So notice that it highlighted the paired reads then when we did a right click, got some options here, and then we can go to mate. What it did is it just highlighted the two mates in red. Okay, so we can continue scrolling along here. You'll see various colored reads as we go along that indicate various things, including the fact that reads map to different chromosomes. So here, let's see what this one, if we left click on that read, we can see that its mate is 657. So I think that this red color indicates that the read is further apart, okay, than are expected. So if we maybe go to mate, now these are the two mate pairs, but this is further apart than a pair of mates is expected to be mapped, okay, based upon the overall insert size distribution that was detected by IGV. Okay, so there's some other things we can do here, just preferences. There's a lot of settings here, tracks, how we handle alignments or visualize them. We can upload charts, various things here. You can look at the preferences. As part of the homework, I have you starting to do a little bit of that just to get a sense of what IGV can do. Let's also do one other thing. Let's load from file the VCF file for the SNPs that I had you call. Here's my call set here. So here, notice how it added a track. I just have a file called callsnips.vcf for this individual. And here what we can do is sort of scroll along. And what you can see here let's scroll all the way in, try to zoom in over a particular SNP call here. So this is in my VCF track, there's a SNP. And I'll let you sort of figure this out in your homework. I thought by default that we would see the bases that correspond to the alleles that are contributing to that SNP call. That maybe it's in the preferences, but I'll let you play with that because I have you, looking at this in the homework. So in any event, you can have it display all of the base calls associated with the reads, and then you can evaluate whether or not that's a real SNP or not. So if we continue to scroll along here, we'll see presumably more SNPs, particularly in regions where we have a lot of reads mapped. The white reads here means that the mapping quality was 0, so they're actually multiply mapped. And here you can see additional SNPs sort of popping up here. Okay, so that's really all I wanted to go over with you. I just wanted you to get a sense of it so it's not so foreign when you begin working on your homework. I think you'll have fun sort of playing around with this. And more importantly, this really gives you a way of quickly and easily doing some sort of just data staring, some quality checking, some familiarizing yourself with the raw data, which can help you to identify possible conditions or circumstances which you weren't aware of about your data set, which in the end can lead to better analysis and better data sets or SNP data sets that have higher quality calls. So we'll also be coming back to this for RNA-seq because what we can do with RNA-seq is actually stare at some of the genes that we think might be differentially expressed, and we can look to see if the read mappings are good and where in the genes are the reads mapping to, which could give us indications of which splice variants or alternative splice variants are being expressed and might be contributing to the differential expression, et cetera, et cetera. But so this toolkit here is very useful, not only for DNA sequencing, but we'll find that it's useful for visualizing RNA-seq data. Finally, here I'll just quickly mention that this is also a set of tools here in IGV that allow us to generate high-quality figures for publication. So if we want to illustrate a data, say a pileup of reads for a gene, IGV has a lot of additional features that allow you to sort of generate publication quality figures for pileups and integrating multiple tracks where you want to see lots of different types of information all together in one figure. We can generate that here. Okay, so I had a couple other points I wanted to raise. Here's a case where we can view paired reads as split screens. So in cases where say two reads map to different chromosomes, which could indicate a translocation event, or to different parts of the chromosome, which could indicate there's an inversion or something like this. We can view that in a split screen. So I thought I'd tell you about my experience with this. This is actually a project that's now published where I was working on with a colleague on Drosophila data, and they had generated a lot of mutation lines that they knew had major chromosomal translocations and in some cases inversion events. They also had large deletions. And so, we were having a hard time predicting these and identifying using sort of the standard pipelines that people have developed to try to identify translocations or et cetera. So we'll be talking about some of this next week, but just in anticipation of this structural variation talk, what we actually ended up doing is they had mapped the mutations. They knew where they were roughly. And so what we just did is we opened the data up in IGV and we started looking for colored reads that indicated for the translocations rather, where piece of a chromosome had actually moved to another chromosome, we started to look for cases where, things were mapping in anomalous ways by looking at the colors of the individual reads. And sure enough, what we started finding was right where they thought the mutation was, we'd find a set of colored reads that indicated a huge number of or anomalously mapped reads. And that was a tip-off. And for each of many different mutant lines, we were able to discover, the source mutations simply by viewing the data in IGV. So, anyways, maybe we'll get to talk about that some more later in the structural variant part of the course, which is coming up, I believe, next week. And here's again just some of the colors. White is unmapped, blue is small inserts, red large inserts or unexpectedly large inserts, and then others, which I was just describing. Finally, I just wanted to come back to something that I noticed some of you were having problems on homeworks, and maybe I could preempt sort of future problems that you might be having. I introduced some of these multi-line command lines, and I think that may have thrown a loop for some of you. You can always type a command line just as a single line, and it can be very, very, very long. In fact, as long as you want it to be, except that it becomes really difficult to read. So to get around this, we do use these backslash here. So we have the end of our command, a white space, then the backslash. And then that indicates that, to the shell that, this command continues on in the next line. So all of the lines, except the last line, must have this backslash. And then you want to be careful not to include white space after the backslash, because that will also cause your command line to crash. So finally, just as a reminder, arguments sometimes have white space in them, particularly when you're, say, grepping for a pattern that has white space. You would have to include that white space in double quotes. So just a couple of commands and things that I've seen some of you guys struggling with a little bit. So hopefully that note will help. So finally, I guess I had a couple more slides on IGV here that I could point out to you. Remember, we can add additional tracks. Here's a case where we have a GWAS. This is a genome-wide association study where the individual points represent SNPs that are associated with a particular trait, and the y-axis here is the LOD score. You'll see these are called Manhattan plots, and these Manhattan plots, because they look like skyscrapers jutting up, are ways of illustrating where in the genome there are statistical associations between SNP variation in a population and trait variation, sort of phenotypic variation. And so you can display this kind of information in IGV. And again, here they've also included gene density. So where along the genome, are there concentrations of genes? So this type of information can also be incorporated, and you can make quite sophisticated versions of sort of graphs in IGV. So with that, I think that's essentially all that I wanted you to be familiar with. This is the information you'll need in order to get through your homework for this week. Once again, trying to keep track of the message board, so keep posting there. Send me emails if you have special problems, or if you need an exceptionally quick response. Usually email is the quickest way to reach me. Okay, so with that, I'll see you next time.